Hi, good evening. This is Dr. Sonal Malhotra reaching out to you. We have a small presentation on infection, types and varieties, and its relevance to COVID-19, in particular with respect to infection control. What is infection? Infection is nothing but an outgrowth of microorganisms within a body. Typically, a body is able to handle uh, uh, growth of microorganisms within itself by having an immune system. However, sometimes, on rare occasions, microorganisms are able to establish a colonization within a host, and this increase in number or colonization is what results in an infection. There are four types of infection. First is based on the kind of microorganism we are dealing with. So there is a bacterial infection, there is a viral infection or a virus-based infection, a fungal infection, and a protozoal infection. There are some more important terms like infectious agent. This is the microorganism that
It's
causes
9 o'clock.
disease. It is called a pathogen. And the disease is caused in a body or a living organism, which is called the host. Both animals and plants can have pathogens, and consequently they can play hosts to different pathogens. Now I'm going to give you some examples of different infections. A bacterial infection. The most common one that we are going to talk about today is Pseudomonas aeruginosa, which actually causes something called an opportunistic infection that we will see a little later. Then viral infection, some examples are HIV, SARS-CoV-2, the ongoing pandemic, herpes virus infection. Then some other infections include infections such as fungal infection by Candida or yeast. Then protozoan infection I've taken as an example, the malarial parasite Plasmodium. There are some different forms of infection. One is called the acute infection. This is a typical infection where an infection occurs in a body and then it is able to be maintained by some sort of medical intervention, or it just disappears on its own because the body is able to take care of it. Such kind of an infection that doesn't last more than 6 months is called an acute infection. On the other hand, a chronic infection is one that starts off slowly um, and builds up over the years, and its symptoms and uh, morbidities increase over the years. One example is HIV. Another form of infection is called what is called the opportunistic infection. Typically, some hosts will not get infected by certain opportunistic pathogens like Pseudomonas aeruginosa. Only under certain defined conditions such as immune compromised state, or because of severe injury due to burns or something like that, do opportunistic pathogens such as Pseudomonas uh, invade and infect human bodies. So this is a good classical example of an opportunistic infection. They normally will not get infected. Only under uh, increased morbidity of some sort or disease state do humans get infected by opportunistic pathogens. So then we come to a very specific kind of opportunistic pathogens, which are called nosocomial infections. These are infections that are acquired in a hospital. Now you would think, why would that be? I mean, you're going to the hospital to get well. Well, sometimes what happens is patients are in there for different kinds of procedures, such as dialysis or something else, and other things like surgeries and implants and things like that. And during those medical interventions, certain procedures are done to them that bacterial species that find a stable environment living in a hospital can actually infect. So there are reservoirs within a hospital that can actually harbor opportunistic pathogens like Pseudomonas aeruginosa, which can live in several ecological niches. So because it has such a diverse genome, it's able to have so many metabolic pathways that can actually virtually reside anywhere, including distilled water. So such opportunistic pathogens are able to live there. We typically accept that 48 hours post entry into a hospital, any opportunistic infection that arises will be classified as nosocomial infections, because prior to 48 hours of entry into the hospital, it is possible that the person walked in with that infection. So that is one of the reasons why we do that. One of the classical examples of an opportunistic pathogen is Staphylococcus aureus, which is a gram-positive opportunistic pathogen. Typically in humans, it doesn't cause any significant problems, but under comorbid conditions, it can be very life-threatening. It's a major cause of hospital and community-acquired infections. What are community-acquired infections? Those are infections that are acquired within a community in a large gathering, such as a gym or a men's locker room or a swimming pool, a public swimming pool. What happens there is people sometimes come in close contact to each other because they share locker spaces, or they share the local bathroom within the facility. In such a case, what happens is people may exchange some things that allow for transmission of a pathogen from one person to the other, and these are called community-acquired infections. 50% of all the hospital-acquired bloodstream infections and 30% of endocarditis and 20 to 25% of pneumonia are caused by Staphylococcus aureus. Similarly, about 30% of people are colonized in their nasal passages by Staph aureus, and out of them, 1% are infected with MRSA. What does MRSA stand for? It stands for methicillin-resistant Staph aureus. So this is a bacteria version of Staph aureus that is resistant to the antibiotic methicillin. And so consequently, you can see eradication of this using a methicillin-based regimen would be very difficult, and alternative medications need to be used. So Staphylococcus causes, as you know, a lot of hospital infections, and about 13% of the 2 million cases of hospital infections are caused by Staph aureus, which results in a death rate of about 60,000 people out of the 2 million. So it's quite a significant amount, which needs to be looked at. Now we're talking of some different kinds of nosocomial infections and the percentages of bacteria that are found in there. So there are different nosocomial infections that have different routes of entry. So there are some things called bloodstream infection. So among all the nosocomial infections, bloodstream infections form 18% of those. Within the 80%, coagulase negative staph form 31.8% of those infections, Staph aureus 13.6%, and Pseudomonas 4.2%. That's how you would like to go through this table, understanding that among the nosocomial infections, bloodstream infections account for only 18%. And then among the 18%, you have these different bacterial players or species that are responsible for those bloodstream infections. Similarly, surgical stream infection refers to surgical site infection, which accounts for 10% of all the nosocomial infections that occur. The rest being pneumonia and urinary tract infections and other things, implant-based infections, etc. Enterococci, coagulase negative staph, Pseudomonas, and Staphylococcus are the major species of bacteria that result in surgical stream infections. Now here are two slides that show three different colony morphologies of Pseudomonas. One being the typical mucoid version that we see in CF isolates or cystic fibrosis isolates. So this is the mucoid variety, which has got this exogenous mucus made out of an exopolysaccharide called alginate. Then here on this other plate we have regularly shaped colonies that are the size of about a colony of E. coli. And then we have some really small colonies here that I have to kind of strain and look. Those small ones are called what are called the SCV or small colony variant. Now all of these have now been analyzed at the level of their genome. So, and a lot of genomic variabilities have been identified in these different colony morphologies. They also have, of course, altered physiology and growth properties. Now we had talked about bacterial growth as being a liquid phase culture, but Pseudomonas and Staph aureus, among other bacteria, can also grow on solid surfaces and form very complex structures called biofilms. And biofilm-based infections on implants and all that are also another important source of nosocomial infections. And frequently we try to identify genes that are involved in formation of biofilms. So here you can see a section of a 96 well plate that was inoculated with different strains. 8325 is a typical prototypic strain of Staph aureus. Newman is also similarly another strain of Staph aureus. And then we have Staph epi, which forms very good biofilms. And then we have a Staph epi mutant in the M10, which is located in the ICA locus. And this ICA locus is involved in biofilm. So when you knock that out, of course, the ability of Staph epi to form biofilms is completely abolished. So what we have done here is grow cultures for 12 hours, remove those cultures, wash these plates, then re-inoculate with media and leave overnight, and then remove the media, wash the plates several times, and then stain for bacteria that are growing on the surface of the plastic by staining with crystal violet, which binds to protein and DNA. So all these blue splotches that you see are bacteria that are growing on a solid surface, presumably being biofilm then. So one way to visualize is qualitatively by looking at this panel and finding out strains and mutants that actually grow in this condition. So we have this 6A5 mutant that, as compared to the wild type 8325, shows much reduced ability to form biofilm. So we can also extract the crystal violet and then measure the blue color at an optical density of 595 nanometers in a spectrophotometer, and then plot the graph here to have a more quantitative idea of how robust the biofilm is. Here is another biofilm. In this case, it is Pseudomonas aeruginosa growing on aluminium coupons made from aluminium foil, and the biofilm is allowed to grow for 12 hours at 37 degrees centigrade and then washed and stained with crystal violet. And you can see that the bacteria do actually grow. So bacteria growing in biofilms have actually, the reason why we study that is because they have an intrinsic ability to change their physiology. One of the most important aspects of that is an resistance to antimicrobial peptides and antibiotics. So when you have biofilm forming cells, then those are already naturally resistant and therefore hard to eradicate, and also hard to visualize and treat in any other way. Once they establish an attachment on a solid surface, they can also move from the solid surface by actually breaking off from that biofilm and moving across the entire organism or host and re-establishing a colony somewhere else. So I have here highlighted in red indwelling devices like dialysis catheters, prosthetic joints, long lines in oncology, then central venous catheters, urinary catheters. These are the kind of implants and implantable devices that are frequently colonized by bacteria and forming biofilms. So they are important cases to study for infection control in some way or the other. Now here is a look at the genome of Pseudomonas aeruginosa. As you can see, it is very extensively mapped, and then homology scanning studies have been done to identify function based on structure alone, and also from other research studies. So by having all this genetic information, we are able to identify genes that may be involved in biofilm formation and identify targets for drug design and also antibody mediated inhibition. So we can inhibit by antibody. We can also have expression data that shows when those genes are expressed in biofilm formation and use the same data and resources to identify new targets for breaking down biofilms. So this increased knowledge of bacterial genomes and expression studies will help with diagnostic because you have the genetic data, so you can identify regions that are important for biofilm for forming very unique structures or unique areas that can be used for diagnostics and profiling studies. Now let's move to infection control. What do I mean by infection control? Well, once an infection has been established, we have to figure out ways to stop that infection from growing further or trying to reduce it in one shape or the other. And here we come down to talking about effects with respect to COVID also. So you'll see some of those things. So antimicrobial agents could be several things. They could be antibiotics or antibiotics, and then sometimes when you use antibiotics, you get bacterial isolates that exhibit resistance to the antibiotic and do not grow or are unable to die in response to that antibiotic. Then we can also use disinfectants. Disinfectants are agents that are used to sterilize non-living surfaces like tables and chairs made from steel or plastic, laptop computer keyboards, mobile screens, and currency notes even, because those can get infected. In particular, there has been a study now to show that COVID-19, the SARS-CoV-2, can actually live on currency notes and be infectious for up to 28 days. So most of the time, 70% alcohol works very well to sterilize to up to 95%. And also, we have found now recent studies that show use of betadine along with alcohol mixed. Those are even more active. Antiseptic usually refers to some compound that will actually grow and eliminate bacteria growing on the surface of your skin. So you can use chlorhexidine, or you can use hydrogen peroxide, or you can use alcohol as well, and even betadine. In fact, betadine has been shown to be used to irrigate surgical wounds also. So it is actually can be used in a little more invasive manner. And more recently, betadine has also been used to in one simple clinical trial study where they have looked at injecting betadine or spraying betadine into the nose and mouth area in SARS-CoV-2 positive patients, and showing that in 3 or 4 days of such treatment, 4 times a day, they can eliminate infectious virus particles. So they can eliminate spread potentially. Antibiotics, on the other hand, are used clinically for internal targeting of microorganisms that are growing exponentially within the body in an uncontrolled manner, and to suppress their growth so that the body can fight it and get rid of the infection. The best strategy for COVID-19 so far is social distancing, mask wearing, and sanitization of self, and plastic and currency notes in particular. So I would recommend using an N95 mask. I personally use a close-fitting 3M N95 mask with an outer 3-layer cloth mask, and sometimes I will use a face shield when I have to go to a store where I know social distancing is going to be difficult. So here's a picture of an N95 mask. It has got this nose clip that when you put it on your face, you can press it so that it forms a very close fitting surface to your nose and nose bridge, and then you have these yellow elastic bands that are there, and they stretch tightly across your face. So this thing forms a cup-like thing on top of your mouth, and it's very close fitting and reassuring that it will not allow air and consequently virus to go through. So what I've shown is that 0.5% betadine from some studies inactivates the virus SARS-CoV-2 in 15 seconds to up to 30 seconds. You basically lose complete titer. So when you do a virus titer dilution assay to determine infectious particles, this betadine treatment pretty much kills 99% of the virus. 70% alcohol also does, and we can use 5% betadine with 70% alcohol and hope for a synergistic action. I haven't measured it, but we have been using this for the last 1 year during the pandemic, and I think it's working since we are okay till now. Social distance of 6 to 10 feet, because up to 10 feet you can isolate virus from COVID patients. Hand sanitization, mask wearing. Then on top of that, if you have been infected, you can home isolate and contact trace people whom you have come in contact with, and use mobile tracing apps to help identify other people at risk and to people from whom you may have got or to whom you would have passed the virus. In addition, lockdown strategies and limited movements like limited exercise times outside also will help this thing. The mask can be sanitized with betadine. Here I have some images of how the virus actually enters. It attaches to the surface. Being an enveloped virus, it attaches via the spike protein to the ACE receptor and then internalizes into the lysosome, and then the virus genome is released and stuff like that. This is a structure of the ACE protein. I'm gonna stop here because I'm going over time and just show you pictures of how I mask. This is my first mask, the second cloth mask, and the face shield, and that's how I look when I do that. So I will stop here and thank you everybody for the time, and I look forward to hearing from you about any questions and any queries. Thank you very much. Have a wonderful day.